everyone, welcome to Learn Science with Neha, a channel which is going to make your science more interesting, interactive and colorful and is going to provide you with ready handy notes too. So what are you waiting for? Let's start with Fundamental Unit of Life, chapter number 5, grade 9. If you talk about a cell, what comes in your mind first is a cell having a cell membrane. Cell membrane will demarcate a boundary and will separate a cell from the external medium. A nucleus, which is a control house of the cell. Cytoplasm, which is like a jelly-like material in which all other organelles float. Nucleoplasm, which is like a liquid present inside the nucleus. And together nucleoplasm and cytoplasm, you call them protoplasm. Along with it, if you go deeper into the nucleus, there is a nucleolus, which is a dense matter made up of RNA, and RNA is going to make proteins further. Now comes who discovered cell. It was a scientist in 1665 by the name of Robert Hooke who took out the bark of tree and put it under the microscope, even which he designed himself and called it Hooke's microscope. And he could see chambers inside. He said, is it a honeycomb? No, it can't be. But he called all the small chambers which you see here as cell. From there, the word cell was coined and we're using it till today. But the tragedy was he could only use dead cells, which was the bark of a tree. Then came a scientist by the name of Anton von Leeuwenhoek. He was a scientist who in the morning after 8 years, he just grabbed the cheek cells of him and then put it under a new microscope which he discovered, which is Leeuwenhoek microscope in 1673, and was surprised to see there were so many things moving. He said cells are not always dead, cells are living too. And then the journey of microbiology started, and then he started working on WBCs, RBCs, yeast cells, bacterial cells, sperms and everywhere. He used to take water from the pond and kept it under the microscope and could see many cells were moving. Thereafter, many organisms were also formed. We discovered so many things around. Then came these three scientists by the name of Sheldon, Schwann and Rudolf Virchow. Sheldon and Schwann, they were the scientists. Sheldon worked on the plants, Schwann worked on the animals and gave the same statement that plants are made up of cells, animals are also made up of cells. But Rudolf Virchow said, although everything is made up of cell, but all cells came from pre-existing cells. That means if we start our life from a single cell by the name of zygote, then this cell only divides to give 2 more cells. This cell only divides to give 2 further more cells, and then the cells keep on multiplying, but the origin remains the same, like this. This cell originated from here. This cell originated from here. So nothing is new which is being formed. Only from the previous cells, new cells are formed. So this was called as cell theory, which said all living things are made up of cells. Number 1, it can be one cell in a unicellular organism. It can be more cells in multicellular. Second point was the cell is a basic unit of life and new cell exists from pre-existing cell. That is a cell theory, and you will mention the name of these scientists here. Then came the dyes. When the plant and animal cells were formed, then came the challenge that how can we see every single organelle under the microscope? Then we discovered number 1 was the glycerine, in which we initially used. Glycerin we used to stain the organelles because glycerin can retain moisture and it will not dry up. Irrespective of water, if you see, water will dry up after some time and the stain will be, and your slide will be useless. But glycerin will retain the moisture and you can use it further. Plant cell was stained with safranin, you can see red color. Animal cell was stained with blue color, which is called as methylene blue. And then comes a question for you. If you dye a cell, which of the organelle you will get prominently visible in the microscope? Give me the answer in the comment section below. Now came the name of scientists. Number 1 was protoplasm, was given by Purkinje in 1839, and Robert Brown was a scientist who discovered nucleus in 1831. So you just have to remember name of these scientists. And then came cell differentiation. That means we are multicellular. In unicellular organism, if you see, one cell is only performing all the activities, may it be ingestion, digestion, excretion, respiration, circulation, everything, because one cell is independent. But in our body, we have different systems like circulatory system, respiratory system, digestive system. So we have got different kind of cells which are responsible for performing their activities, which are apt to that system only, which are designed for that system only. So this is called as cell differentiation. You can also call it as division of labor. So depending upon shape and size of the cell, based on their functions, they are categorized and they are allotted different duties. Like red blood cells for transportation, as they can carry oxygen, and red cells also, hemoglobin. Smooth muscle cells, because of contraction and relaxation, they go in the muscles. And then nerve cells, they go in the nervous system, as they can transmit the nerve impulse. And then came structure, that is, we all start our life from cell, then cells divide to form tissues, tissues to organ system, organism. So this is what differentiation is, and we have got different shapes of cell and different sizes also. To your surprise, the largest cell is an ostrich egg, which is around 170 millimeter big into 130 millimeter broad. This cell is so big that even if you stand on the cell will not break. I have tried it. Now, if you talk about a body, the longest cell is the nerve cell. Largest cell in human body is an ovum, ova, and the smallest cell is your sperms. The bacterial cell, which is even more smallest, it is only 0.1 to 0.5 micrometer. Next question for this session. How many meters will make 1 micrometer? You have to give me answer in my comment section below. Now, if we talk about the organelles, you will see cell membrane. Cell membrane is a living part of the cell which is made up of lipoproteins. If you break this lipoproteins, it will be lipids and proteins. So here I have written lipoproteins. So lipids means fats, protein means proteins. So fats and proteins combine to form lipoprotein. Now, if we talk about cell membrane demarcates a cell from the external medium. Number 1, it is present both in plant cell and animal cell. Number 2. Number 3, it is selectively permeable. That means it will allow only few material to enter inside and move out, and not everything. Number 4, if you talk about two terms, diffusion and osmosis. Through cell membrane, gases will diffuse, oxygen will move in, carbon dioxide will move out. At the same time, liquids are also moving in and going out. So exchange of gases occur by diffusion, whereas exchange of liquid occur by osmosis. Now let's do osmosis in a bit detail. Due to osmosis occur in three type of solution. Number 1 is isotonic solution. Solution means solute and solvent. I have taken red color potato here. So potato inside is also having liquid and salt, and outside I have put it in a medium which has water and salt. If concentration inside is equal to concentration outside, concentration means combination of salt and water, inside and outside is same, then the amount of water which is entering inside will be same as amount of water moving outside through the membrane of potato, and the size of potato will not change. So this kind of solution is isotonic. But if I put a potato in hypotonic solution, that means outside is just a water, very less of salt, but inside it has lot of salt. So water will always move from higher concentration towards lower concentration in osmosis. So that's how water will go inside and the potato will swell. But if I take potato in the hypertonic solution in which salt is more, Hypertonic means salt is more, water is less as compared to inside. Then water from inside will start moving from higher concentration towards lower concentration of water, and that's how the potato will shrink. So this is how you can see the size difference if you are putting a potato in different kinds of medium. And then comes the difference between osmosis and diffusion. If you talk about osmosis and diffusion, if you talk about diffusion, diffusion can occur in any of the medium, may it be solid, liquid, gas, but osmosis will occur only in liquids, number one. Number two, movement of particles will be random in diffusion. That means particles, if you see, they are moving here and there without any particular direction in this case. So here zigzag movement, for example, I am applying a perfume and you can smell the perfume in the next room also, because the perfume particles, which are very volatile, they will start moving randomly here and there and you can smell it. But in case of osmosis, it will occur only from higher concentration towards lower concentration of liquid and through semi-permeable membrane, which is not required in diffusion. So these are the points of difference between osmosis and diffusion. Now comes the cell wall. Cell wall is not having any membrane. Cell wall is cell wall. So cell wall is very, very rigid, very supportive. It is only present in plant cell. Why not in animal cell? Have you ever thought of it? Reason is, plants are not moving. Plants will stay wherever they are. You have got big trees and outside sometimes it's very hot, sometimes very cold, sometimes it's raining. But plants cannot rush here and there. So that is why they have got a protective coat by the name of cell wall, which protects the cell of the plant from adverse conditions. But in our case, we can protect ourselves by moving, since we have hands, we have legs. So that's why we don't require cell wall, we just have cell membrane. Then comes cell wall is living or non-living. It is non-living component which is made up of cellulose. And then comes very, very important point, plasmolysis. This is a term which is used only and only for plant cell. So the case is, if you have put a plant cell, I am making a cell wall and I am making a cell membrane. If you have put a plant cell in a hypertonic solution, okay, hypertonic means salt is more, water is less. So water from the cell membrane will start moving out in the outside medium, and slowly you will see cell wall will not change its shape because cell wall is non-living, but cell membrane will contract and will go till this level. So that means plant cell has undergo shrinking process. This is called as plasmolysis when the plant cell is placed in hypertonic solution. And the next property of plant cell wall is it is completely permeable for liquids and gases. No matter how much amount is coming, so liquid and gas will keep on exchanging without any stoppage, unlike cell membrane, which only allows selective material to enter. So it is completely permeable. Cell wall and cell membrane selectively permeable. Now comes the question time. So number one, there are two cells, cell A and cell B. You have to identify what is a kind of cell. Let's give a try. The first cell is in a good condition. So can I say it's a turgid cell where water amount is too much and it is putting a pressure on the wall, and although both are plant cell only having double membrane, so I can call it as a turgid cell, whereas this cell might have been put in hypertonic, so it has become a plasmolysed cell. Next, what will happen if cell B, which was plasmolysed, is placed in hypotonic? Hypotonic means water is more, salt is less. So it will undergo endosmosis, means water will start moving inside, endosmosis, and the cells will regain its original shape. And if cell A is placed in hypertonic solution, it will undergo exosmosis. That means water will start moving out and it will slowly undergo plasmolysis. So here it is endosmosis. You can write these terms as well. Now comes, yes, We are right. You can check the answers. Next is, what will happen if excess amount of fertilizer is added to green grass? Fertilizer means salt. So if salt is more around the grass, so water from the grass will start moving out and your grass will die after some time. Second question, salt is added to cut raw mangoes. So you might have seen a lot of salt is added when you're putting mangoes because salt is creating a hypertonic solution, and that's why the water from the mango will start moving out. Mango will be preserved for a longer time without any bacterial attack. Now comes cytoplasm. Cytoplasm is water plus salts present in the cell. It is a medium in which all other organelles float. So I can say cytoplasm is like a sea and organelles are like your boats and ships and cruises which are moving in the sea. So jelly-like material in the cell is your cytoplasm. It provides medium for transportation. Organelles can float. Transportation of glucose, gases, waste material, food material, everything. Proteins. So it has got 80% of water and just 20% of salts and hormones, cells, enzymes. So now comes the nucleus. Nucleus is a control house of the cell. Nucleus is a double membraned organelle. Not all organelles are double membraned. Nucleus is double membrane. So nucleus has got small pores from which materials will be exchanged. And now I am going deeper inside the nucleus. That is, after the nuclear envelope, which is double membrane, nucleopores, you have got these chromatin material. Chromatin are random thread-like structures of chromatin only. You will not say exactly chromosomes. So they will combine to form chromosomes later on. And then if you go still deeper, you have got nucleolus. So nucleolus is a body in which RNA is present. And RNA is responsible for production of ribosomes, and ribosomes will create proteins in the cell. So nucleus is almost, you can say, autonomous body, semi-autonomous body, because it has got its own DNA, chromosomes and proteins also. So let's read this nucleus. I told you it was discovered by Robert Brown in 1831, and it is one of the largest organelle which occupies 1/10 of the volume of the cell. It has got nuclear membrane, it has got nucleoplasm, which is a fluid in which this chromatin is floating. It has got nucleolus, which has got RNA, which forms ribosomes, and chromatin, which are thread-like structures and contain DNA. DNA is a genetic information, contains genetic information in the form of genes. Now comes very, very important question as per exam point of view. What is the difference between DNA and gene? If you see here, you can see two strands are moving. So these two strands are made up of DNA, and this ladder, if you are seeing, that is a gene. So gene is made up of set of 3 proteins like this. So this is protein 1, protein 2, protein 1, protein 2, protein 3, protein 4. They are random proteins. So set of 3, triplet you can say. This triplet will form one gene. Okay. So these genes, they will combine to form a DNA. So this, if I see on your right side, so this whole is DNA, and these are the set of genes which are formed. And DNA is moving towards to form a condensed structure which is called as a chromosome, which undergoes division at the time of reproduction. So gene, I am reading it out now. So gene is a sequence of 3 proteins that refer to any specific characteristic in offspring. DNA are carriers of genes. DNA carries the genes. DNA combines to form chromosomes, I showed you. So how will you remember it? DNA is a long string like this. Genes are small knots, and chromosomes are coils of these knots together. Denser, denser, and denser. Now comes the next part. Prokaryotes and eukaryotes. So prokaryotes are the primitive cells which were formed when Earth came into being, and slowly and gradually they were advanced and they turned to eukaryotes. Now let's read out. You have got some bacteria, which is a prokaryote. Prokaryotes do not have a well-developed membranes. Even the nuclear membrane is not present. They don't have a well-defined nucleus also. They have got a nuclear region, which you call it as nucleoid. Okay, so this is a prokaryote. You can see they only have ribosomes. Okay, if you get a labeling, ribosomes are not having any membrane. So this is the only organelle which is not carrying any membrane in our cell, that is ribosome. So ribosomes are present in the prokaryotic cell like this. It is common, and along with it, it has got a nucleoid, and rest you have got cell wall, as bacteria has cell wall. Okay, along with it, it has got flagellum to change the direction. But in eukaryotic cell, you have got all the membrane-bound organelles, where the nucleus, ribosomes are common, mitochondria is there, cell membrane is there, and so on. So most eukaryotic cell prokaryotes are unicellular, eukaryotes are multicellular. Nucleus is poorly developed or absence of nuclear membrane. Nucleus is well developed, nucleolus absent, it is present here. Organelles are absent, organelles are present here. Example is bacteria and blue-green algae. This is very important question, you must know. Now comes, let's check our knowledge again. So this is what you are seeing on the screen. I am sure you all know it is a nucleus. So nucleus, and what is X? Dense structure inside the nucleus is nucleolus. And what is Y? Y is chromatin. What is the function of X? It is to carry synthesis of RNA, ribosomes, and ribosome will create proteins. Now, that's it. Endoplasmic reticulum. Endoplasmic reticulum, for example, I have to move from one place to another. I need a road. Same is the case. This ER will provide a framework to the cell. Framework to the cell means like it will start from nucleus and it will start moving from nucleus till outside. So endoplasmic reticulum is a dense framework on which all other activities are happening. May it be protein synthesis, may it be digestion of foods, and may it be transportation. Everything is happening here. So let's read out. Number 1, it is a network of membranous tubules with cytoplasm of eukaryotic cell, continuous with nuclear membrane. So it starts from nuclear membrane and goes in the cytoplasm in the form of a network. It has some structure as plasma membrane. It is involved in protein and lipid synthesis. So ER is of two types, rough ER and smooth ER. Rough ER has got dot structures which are ribosomes attached to it, but smooth ER don't have ribosome. So smooth ER will synthesize lipids, whereas rough ER will synthesize proteins. Now, along with it, as I told you, it serves as a channel through which proteins can be transported to different regions in the cytoplasm. Along with it, I told you, it forms a framework in which all biochemical activities and reactions can occur. And one very important function of smooth ER is it detoxify the liver. Detoxify our food. So it is very important organ and the function is performed by smooth ER. Now is biogenesis. Since I told you smooth ER forms lipids, rough ER form protein. So smooth ER and rough ER will combine their product and they will form cell membrane. And this cell membrane is transferred to different organelles of the cell. So that's it. This is part 1. Hope you liked my video. Hope it is useful for you. Do like, share and subscribe to my YouTube channel and press the bell icon to get the regular updates. Stay tuned. Thank you. Bye-bye.